演示。最后实验完成后是一个问答环节。整个过程中大家有什么问题可以结束后跟陈博士讨论。现在我们就把时间交给陈博士。Hi, Dr. Chen. Can you hear me?
Hello.
Yeah.
Hello, everyone.
You can start.
Yeah. Thank you. Thank you for nice introduction.
Okay.
Yeah. Hello, everyone. Yeah. Thank you and welcome to our virtual workshop about microfluidic today. My name is Ching, and I'm PD in chemical engineering, and I'm an application specialist of Dolomite in Asia Pacific regions. So today we will be talking about the microfluidic microfluidic technology and its application for using for production of the liposome for controls drug encapsulation. So today workshop will including the 3 sections. So section 1 will be a presentation, and the section 2 will we will run live demonstration with our system. Just you can see over there. And the final section is with discussion and questions. So before starting the workshop, so I'm very appreciate if everyone can mute your microphone, which can help us to avoid a noise, unwanted noise. And okay, so let's start with section 1. So I will share my screen to share to everyone to my presentation. So everyone can see my screen now?
I can see you.
Okay, good. Yeah, okay, yes. So let me start the section 1 with a presentation about microfluidic production of liposome for controlled drug encapsulation. So the content will include including the introduction about Dolomite and microfluidic technologies, and liposome particle production, and next is the system scale up and the system flexibility and other particle formation, and finally is summary and conclusions. So Dolomite is a part of the black chain group. So we develop, design, and sell market-leading product through our global leading brand, including the Dolomite. We provide cutting-edge product for microfluidic. The particle work, we provide the precision engineered nano and micro materials. And the Dolomite Bio, we provide innovative product for high output single cell research. So we have more than 100 people globally with headquarters in Royston, United Kingdom. And we have office in the United States, Japan, and in Vietnam for supporting the customer in Asia Pacific regions. We also have distributor in more than 25 countries that can help all the customer in the world with free support from us. And Dolomite particle engineering team is a team of the chemists who, I don't know, everyone can see my next slide. Okay, it's a little bit large. I don't know the region, but okay. So let me start, continues. So Dolomite particle engineering team is a team of the chemists and engineer, so which focus to produce high performance of nanoparticle and microparticles with precision controls over size, shapes, and structure by using the microfluidic and flow chemistry. So we focus on biomedicals and pharmaceuticals industry, as well as we supply the standard product, such as particles and immusion stabilizers. We also advise customer on the microfluidic setup and the processes. And if required, we can undertake the proof of principle project before buying or purchasing the product equipment. So with microfluidic technologies is enable formation of the high monodisperse droplets in range of micrometer to 100 micrometer with very complex structure like emulsion or double emulsion or encapsulated gas bubble or xenous particles which the particle have heterogeneous surface properties, and also the hybrid materials. And also, the droplet can be used as a microreactor to create the complex compounds as the sample can be confined or dilute or mixed or split or analyzed. So here showing just example, a very simple example with in color in the batch reactor as traditional method in nanoparticles generation. To show the limitation of the batch method in particle size distribution. So as you see from the ink colors, there are so many factors can be affected in the process. So that's can generate that's a little the batch methods have a very polydispersed. So in the right slide, so you can see the TM images, real TM image nanoparticle produced by the batch synthesis is showing very wiring side distributions. So otherwise, with the flow microfluidic methods, so because it's flow is flow process, it's continuous process, so it's very rapid mixing, and the particle generation production can be generated by in flow with the nucleation and growth, and very easy to control the parameters like the flow rate, temperatures, and the mixing. So it can generate very monodispersed particles. So in dry images in here is showing the TM images of nanoparticle, growth nanoparticles with very similar condition with the batch methods. Is showing very monodisperse by using flow synthesis. So the microfluidic technology is can be applied in very wide range of application in industries. So it can be applied, for example, in pharmaceutical industries like drug delivery or drug encapsulation. It can be applied also in nutraceuticals industries or cosmetic industries. Or food flavor and fragrance or research academia, which can be applied with R&D process before applied in the industrial. And it also can be applied in biotechnologies with the single cell research or the cells development, or it also can be applied in agriculture and other industry like the materials, refineries, chemistry or aerospace or the hospital as well. So here's showing some example of our Dolomite customer in the world. So we provide our product to very big, very famous institutes in the world, like the MIT or Harvard universities, or some big company like Google or the NASA or pharmaceutical company like Shire. So it's very wide range application. So it means we have very many experience in the supporting customer in the very wide range application. So next, I would like to showing the basic concept for microfluidic technologies. So this one showing the how the microfluidic how the emulsification process can be work in the both traditional methods like batch methods and microfluidic methods. So as you see in here with the batch traditional methods, so the emulsification process works through the input of energy into two immiscible liquids and applies energies like stirring or sonication. So after that, so the emulsification can produce by stirring. So because of the process is too many factors as I saw in previous slide, so it can make very polydisperse sample. And when we do with the encapsulation process by batch method, it's very easy to destroy materials being encapsulated because we apply very high energies with this process. On the other hand, so it contracts, so it in microfluidic droplets pinched with a very small input of energies. So it's so the parameters effect we can easy to control with the flow chemistry. So it can make a very monodisperse particles, and because of very small energy applied with this process, so it's the materials being encapsulated can be stabilized during process. So here showing the way to the method to generate the droplets. So it's a very many way to produce droplet and or emulsion by microfluidic technology. We can do with the segment flow by using T-junction, or single emulsion by using the X-junctions. Like here is very familiar in microfluidic technologies. And we can do with the double emulsion just can form the droplet inside the droplets of the wide side range. And we can do the cell encapsulation by placing one or a number of cell particles inside of the droplets for the single cell or cells research. And also we can do nanocrystallization by mixing of two streams and subsequent isolation of the reagent into the droplets. Thus, the droplets can be used as microreactors. So the nanocrystallization will be occurs within inside the droplets. Or we can make the xenous particles by incomplete mixing of two streams, forming particle with heterogeneous surface properties. We can generate the microparticle or nanoparticle with different properties on the surface. So what do we need for running microfluidic experiment? We need main three things to run microfluidic experiment. So the first is a way to moving the fluid is a pump, and the second is surfactant. Is a way of adjusting the surface tension of the fluid. So we have to use the surfactant to stabilize the droplets or emulsification after process. And the third thing, and also very important, is the microfluidic device where application runs. So it means the microfluidic chip with the strainers. So here showing our pressure pump, the P pump. The mechanism of the pressure pump, which including the pressure chambers integrate with the flow rate sensor and the sensor displays. So inside the sample can be put in pressure chamber, and we apply pressure in the chamber that can push out the fluid flow. We contain a liquid inside the wire inside the pressure chamber out. And we can control the pressure in chamber is maintained and the constant value when we control by the P pump. And after flow out the chamber, so the flow rate sensor will be measures the flow rate sensor and flow rate of the fluid and control the flow rate by adjusting the pressure inside the chamber. So the advantage of the pressure pump compared to syringe pump is we can do very smooth fluid at very small flow rate. Because with the syringe pump, because of the mechanicals of the syringe so action, so it can, if you do at very small flow rate, so it can make up the high fluctuation of fluid. So actually the flow, a smooth flow is very important in the microfluidic as well as the flow chemistry. And the second advantage of the P pump is we can using the valve to disconnect with the fluid with the chip every time. That cannot do with the syringe pump, because when we do with the syringe pump, if we stop the fluid, so the pressure inside the syringe pump will increase higher than and overload the pressure with the syringe pump. But with pressure pump, we can do it, and very easy to control the process without changing any setup. So this one showing the one factor to control the droplet size. So for the droplet size controlling, we can use two ways. The first way is we can use the different chip junction or channel junction size. So with the bigger junction can form larger droplet size, or the smaller, if you want to make up smaller droplet size, we can use the smaller chip junction size. The second way when we can control the droplet size with the same chip is adjusting the relative flow rate ratio of continuous and droplet phase. For example, if we increase the flow rate of the continuous phase, the droplet size decrease. If we increase the droplet phase flow rate, we can increase the droplet size. And if we want to increase droplet production rate, we increase both continuous and droplet phase flow rate. So setting limitation occurs, we will usually it's occurs at the maximum the flow rate in this case. The other factors also affect to the microfluidic process is chip surface coating. It's strongly depending on the what implication process you do. So we can choose a different type of the coating of surface of reactor. So if, for example, if you want to make up the oil in water droplets or emulsion, you should coating with the reactor walls with the hydrophilic coating. That can prevent the contact between the droplets and the reactor walls, so and it can prevent the aggregation or destroy the emulsion. So if the otherwise, so if you want to make up the water in oil, so oil in here is dispersed phase, so you have to do the hydrophobic coating or the fluorophilic coating, so we can prevent the water droplets contact with the reactor wall and prevent and stabilize and have to improve the stabilization of the particles. This one is showing the particle generation with selecting of the nanoparticle or microparticle. So normally the people using the droplet formation method to make the microparticles with the particle size that are bigger than 1 micrometer. If the people want to make up the nanoparticle, we use a different method. We call it flow focusing methods. So in this method, the flow of the polymers contained in stream is focused into the 18Z between the two converging the water stream water stream with the higher flow rate. So as this focus stream contains dissolves, the polymer is stabilized. The diffusion of the solvent out of the focus stream and the diffusion of water into the focus stream occurs, and and is the destabilize the polymer solution. So this will lead to nanoprecipitation of the particle at the interface between G2 fluids, the interface in here, and flushing out by flushing out the microfluid channels by the continuous phase or the waters. So by this method, we can make a very narrow size distribution in nanoparticles production, and the microfluidic synthesis can bring the very many advantages, such as it can be enhanced control fabrication process because it's very easy to control the parameter with the microfluidic, and also it's a great particle yield and narrow size distribution or small average size, and also it's very easy to scale up because we apply the very small energies with the microfluidic technologies. And this slide, I would like to introduce you one of the applications with the microfluidic technologies is the liposomes particle production. So as we know, liposomes are vesicular nano and microparticles which formed by the lipid bilayers membranes in here, which contain the aqueous core inside. So that we can use to encapsulate the drug in both bilipid membranes and aqueous core lipids. So and then delivering the liposome content drug within the body. Also, the liposomes are widely employed at drug delivery system. And here showing therapeutics area covered by liposome-based products. So because the liposome is biocompatible and easy to produce, also it's cationic lipids form an electrostatic complex with DNA. It's for gene delivery or vaccine development, and it's flexible for both hydrophilic and hydrophobic drug, which we easy to encapsulate through the active or passive loading, as well as directly from the flow. And also it can entrap the unstable compounds like antimicrobials, antioxidants, or flavors, and release them at designed targets. And also it can increase therapeutic index, minimizing side effects, and also it's specifically ligand for the active targeting. Here showing the mechanism of the liposome nanoparticle production. So as I introduced in previous slides, we use a microfluidic with flow focusing methods. So in this case, lipid will dissolve in the organic phase like ethanol or isopropanol, IPAs, or even though with the acetone sometimes. And the outer phase or continuous phase will be the PBS solution or water phase. So it's making the lipid particles is formed by using the process of the nucleation and the growth. And the particle size of these lipid nanoparticles can be controlled very easily by changing the total flow rate factor or flow rate ratio, which determined by the water phase flow rate divided to ethanol flow rates. So also for API or drug encapsulation process, so the API or drug can be browsed or dissolved in both continuous phase. For hydrophilic continuous phase with the hydrophilic API, or interface with the hydrophobic, or can be encapsulated afterwards by active loading methods as the very common method for loading API to the liposome nanoparticles. Here showing the flow focusing method setup for Dolomite system. So we use two pressure pumps for liposome phase, droplet phase, interface, and water phase as the outer continuous phase in here. And in this case, we use a 5-input chip which can design for the making of flow focusing stream to generate nanoparticles. So the result showing in here in this graph is showing we can change the particle size by just changing the flow rate ratio. With increasing the flow rate ratios, we can increase the particle size with the very small size distribution. Here showing a single channel system for microfluidics flow focusing methods. So basically we have 3 pumps, which 2 pumps is using for the pumping fluids with water phase and lipid phase, and the other pump will be used for fluxing or cleaning before and after process. And the 3 pumps will be connected with microfluidic chip. We have different types of chip, like the 3 flow focusing chip, or the normal droplet junction chip, or 5-input chip, which optimize design for making a flow focusing stream, or we can make the nanoparticle with the flow focusing method with micromixer chip to increase the output product. And the process is visualized by using the high-speed cameras, high-speed microscope integrated with high-speed camera integrated with microscope. We can visualize the process during experiment. Here showing some results with our process experiment. In this case, we use the organic phase is 10 milligram per ml phospholipid as a lipid source, and which it will add to the 2.5 milligram per ml DDAB in ethanol. DDAB in here with this role is for surfactant to stabilize the nanoparticles, and the continuous phase is using the PBS solution with pH 7.4. So the result showing in this graph, and it can show very stable formation of lipid nanoparticle with a very small PDI, the polydisperse index width, and also the lipid particles were produced continuously with no intervention, and the particle size was successfully controlled in the region of from the hundredth nanometer nanometer to 250 nanometer by just changing the relative flow rate ratio. Here showing the results we use with micromixer chip, our micromixer chip. In this case, the lipid will pumping into the interface in here, and the water phase will be here and here, and flow focusing the stream will be making a chip junction here, and the mixing step will be improved by the micromixer channels inside the chip, and the results the organic phase in here we use it with the same experiment condition with the 5-input chip as the previous slide. So, and aqueous phase also the same, PBS with the pH 7.4. The result showing the particle size was successfully controlled in the region from the 60 to 120 is smaller than the 5-input chip by just changing the relative flow rate ratio with a very small polydisperse index width, as you can see in here. And this slide we're showing the system scale up. with our system scale up, we generate the high output system. We call it telos. So with the telos system, we can increase output by using two ways. The first is increase working in parallels. So, for example, in the micromix, in the telos chip, we can integrate the 7 chip junction channels can be working parallel in the one chip. So it means we can increase the output of product as 7 times compared to single channel chip. And the second way is increase the flow rate through the individual junction. So by integrate with different chip design, we can increase the flow rate with the telos system. That can be increase the output product. So here showing the how the telos work, with the multiple telos clamp modules can be stacked together to build up a system for large scale production. So with the telos system, we can stack up to 10 clamp module with one clamp module showing in this, and we can using the up to 10 and stack up to 10, and using as the one system, we can increase up to 10 times compared to single chip, or 70 times compared to single chip junction. So here showing some experiment results for liposome production by using the telos system. That showing the particle size was successfully controlled in the region of the 100 to 180 nanometer by just changing the flow rate ratio with the small the polydisperse index width. And with this one, just total flow rate, it can be up to 100 mil per min. It's been very high outputs with using telos system. So the next slide is a system flexibility and the particle formation. As our system was designed as a Lego piece, so it means we can integrate cell disassembles or integrate the module together very easy, and apply many wiring application by changing very small thing in the system with the main system. For example, if you want to make a nanoparticle, we use a different flow focusing chip. With the same system, we can make a microparticle with the droplet chip junction, normal chip junction. Just changing some setup in the tubing connection and chip type, we can make different application in one system. Or if we want to upgrade to telos system, we just add one more module with the telos modules with the basic the component system that's already used for IND scale. So here showing the one of the application for lipids using the microfluidic system. So here is Zent liposomes. So in this case, we can make up the liposome in microsphere particles by using the water in oils droplet with lipids dissolved as surfactant. In this case, we make up the lipids were dispersed in the oil phase in here in the continuous phase, and water droplet will be formed as the droplet phase. And during the droplet formation, the surfactant is the lipids can be absorbed on the surface of the droplet and make up the first layers of the lipids, one layer of lipids. And after that, the second step, the water that we the second step is the water that will receive the final vesicle is placed in the second vial. Second vial which contains lipid saturated oils. And it's a pores on the top of it. So in here, showing here the mechanism. So after droplet on the top is a lipid monolayer form at the oil and water interface, we'll be generate the second layer of lipids, and because of the droplets contain water, which usually is the higher density or heavier compared to oils, so it will sink go to the bottom of the reactors, and by this way, the interface was the second layer of lipids sitting, and the bilayers are complete, and the final vesicle are formed. So we can generate the very microspheres, the liposomes product. So the system setup is also very similar to the others. The application like continuous phase will be connected from the pump to the chip by split in two ways, and the droplet phase contain the water in here. It will pump to the interface of chip junction and generate the droplet water in oil which contain lipid as surfactant, and it can be generated then liposomes. And the other particle also can be applied with this single system. For example, like alginase or co-cells, PLGAs particles, or PLGAs microparticles, or also the soft and hard polyacrylamide base, with or also the PLGA is a nanoparticle, and it can be used for very wide-ranging applications like for drug delivery or drug encapsulation or pharmaceutical or cosmetic or food industry and agrochemical industry or screening or R&D process. So summary, so Dolomite can target many key applications in the area of liposome and other particles, like polymeric microparticle or nanoparticle synthesis, and drug or API encapsulation can be also applied with Dolomite system, and others application like the food or cosmetic catalysis, quantum dot application also can be applied. Dolomite product and particle engineering team expertise enable end user to achieve very high performance and reproducibility process. And Dolomite process can be also scaled up to pilot plant, which can produce with a very high production level using the Telos system. So I'm finishing the presentation today with the liposome production and the introduction about our system for liposome production. So if you have any questions, so feel free to using your chat box and writing down the question in the chat box. So I'm very happy to answer you after the section 2, in the section 3 with the discussion and Q&A. But now we will go to a live demonstration with our system over there, and with today with the application of the liposome production. I will stop my screen now and showing the system over there. Can you zoom in the system as well? I don't need to. Yeah. Okay, everyone can see the system over here. Yeah. Is it clear?
Okay. Yeah, it's okay.
Yeah, okay. It's okay. Yep. Okay, so as you can see, the system is the single the Dolomite microfluidic system, which can be used in R&D process in a lab scale. So it's including the 3P pump with the 2P pump for pumping the fluids or making fluids to the Microminer chip. And the third one we use for the cleaning or flushing chip before and after the experiment. So the P pump in here, I would like to introduce the P pump in here. We contain the pressure chamber in here, and we connect to the flow rate sensor. It can be controlled easily flow rate by the adjusting the pressure inside the chamber, and the tubing connecting with the microreactor chip in here. We connect with also the two-way valve. So it's very easy to control or stop and continue the process during the experiment. And here's the Microminer chip, the reactor chip. In this experiment, I will show you the chip using our applications. We use the 5-input chip to make flow focusing methods. Using the flow focusing stream to make up the nanoparticles, so liposome. In here, sorry, I will take the globe in this case. So here, I don't know, can you see this, the chip, microchip sensor? Can you focusing on this? Okay, good. Yeah, can you see the chip channels? Right?
Yeah.
Okay. This is a 5-input chip with the 5 input chip channels. So we contain the 4 input and 1 output way. So the 4 input can be used one for the interface, and for the other thing we can do with 2 different reagents in the continuous phase. One output channels we will continue here in this case, in this line. Okay. So the chip will connecting with tubing by the edge interface and the linear connector, and the process can be visualized by. Sorry, can you zoom in? Yeah. Be focusing. No, it's not focusing. Yes. Okay. Yeah, it's visualized by high-speed digital microscope here. So all the process, the droplet formation or the flow focusing junction can be formed, can be visualized by the high-speed digital microscope. And we're using the software as FCC software. We call it FCC software to control all of the pressure pump, the microscope, and the others module of our Dolomite microfluidic system. With the pressure pump or in here, we can also control the fluid flow rate by using the itself function by changing the manual knob control in here. But otherwise, every equipment the Dolomite system can be controlled easily with our free software. We call it the flow control software, the Dolomite Flow Control Center software. And before starting the live demos, the experiment, I would like to just introduce the here. So Telos scale clamp, Telos module for outputs, high outputs production. So this one is one clamp for Telos, and you connect with the Telos system chip in here, and this one will connect with fluid through the pump in here by 3. Oh. And okay, so this one will be very easy to integrate up to 10 clamps in one system. So to increase the flow rate or the production scale. And if you want to control the temperature, we also have the TCU temperature TCU modules we can control the very fast response and accuracy the parameter from 1 to 100 degrees C or 120 degrees C by using this. So the chip will be connected in the top surface of these modules, and the temperature will be controlled easily. And or otherwise, we can heating or control temperature by we have also the remote pressure chamber, as you can see over there. So it can move and put it into the hot list or storing or to making storing or to make warming up the solution before going to the microfluidic chip. Okay, so that's all of our single channel system. We can be used in INDs or lab scale. So now we will run with the live demos experiment. We can apply to make up the nanoparticle of the liposomes by 5 inputs microchip. So in this experiment, we use a 2 pump. 1 pump contains the PBS phase, water phase. The other pump will be contain the ethanol and the lipid phase. So it's pumping to the chip. So and controlled by flow control center software. I will share my screen again to see how the software works, and let me share my screen again. Yeah. So now everyone can see the screen, right? So here in screen, you can see in here, it is our flow control centers. So it's a free software. So that can be used to control the own of the equipment, own of the module in Dolomite system. So in here we can see in here the 1 pump. We have 1 pump to contain the water. We pump the water inside, and the other pump will repeat with the ethanol. And we can control both of the mode of the pump by pressure or flow mode by just switching two functions in here. Yeah, and this one is showing the screen for microscope imaging in here. So I'm just using. You can see the smaller the resolution of this. So it's okay. Yeah, you can see it's a 5 input chip. So the interface will be here. Interface will be here, and the outer phase will be here and here, and here. I mean the outer phase, yes, and just the sample product will be go by output in here in this way. So in this experiment, we use just only the outer phase, just only water and or PBS solution. So we do not use the two reagents. So we will block this channel. This channel, the outer channel, we block, and we use 2 in the channel here, in the setup. So we will make up the flow focusing flow in here. So I can. It's more clear in here. Okay. So now I will start experiment with just apply the pressure to the water phase, the outer phase in here, and we just adjust pressure. For example, 1000 pressure. So when we enter pressure, so the P pump will adjust pressure to the setting up very easy, very quickly, easy and very quickly here. And we can open the valve from here. We open the valve from here, and we can see the flows, the water already. It's around 500 microliter per minute. And now I also apply maybe 900 example of the lipid interface in here, and open the valve, second valve in here. Yeah, so it's already making a flow focusing stream, so I can zoom in the chip to make sure. Okay, you can see very clearly. Okay. We just adjusting the focusing of microscope. So now everyone can see the flow focusing method here. Everyone can see it, right?
Yes, yes.
Yeah, so I can enlarge. So as you can see in here, so when we can change the flow rate ratio by just changing this one. For example, I increase to continuous phase to decrease the thinner of the jetting, so laminar flow. So in here, so lipid and ethanol will be here, and will be dispersed, mixing in here, and fluxing the nanoparticle. Lipid nanoparticle can be generated in interface between the water phase and ethanol phase, and flushing out by the flow, by the fluid. Here, so in here we can changing both. For example, we can make a slower this here to decrease. For example, like this. Here you can see it's changing. So it's very easy to make investigate or optimize, do optimization of the parameter with the microfluidic method to achieve the right condition for your target liposome production by this method using. So here, this is live demos for the production of the nano liposome by flow focusing methods. So because in lab condition now we don't have any post analyzed equipment, so you can read it from our application note. Just we already published in our website, and you can download it for free. And also maybe Wu, you can contact Wu for Wu is our distributor in China. So you can contact to Wu to get the Chinese version of the application note that we already generated in the Chinese version.
Okay.
Yeah. So I finished the demos live demos for experiment of the nanoparticles. So we will go to the next section is discussion and Q&As. So every have question, please feel free to ask me through the chatting box, so I can check it from here, and we write answer. And if we don't have enough time for your question, maybe you can give me the information, your email, so I can answer you, all of you, try to answer all of you through the email as well. So yeah, feel free to ask me the question.
大家有什么问题可以直接问他，或者把这个问题打在这个聊天这个框里面。
So everyone, in case everyone cannot write out quick question in the chat box, you can speak in the Chinese with the Wu, and after that you will we will translate to the English, and I can give you the answer as well.
大家直接用中文问也可以。
都起来。听得到吗？
Yeah.
能听到。
Okay, it's Xiangxiang Wu. Okay, it's me. I want to know which kind of the surfactant are optional. Okay, it's a very wide range of the surfactants. It's many the surfactant can be used. It depending on the application as well as the lipid source. So we can choose a different surfactant. For example, we can use in our application note we use a DDAB as a surfactant, but with we can also use a sodium citrate as surfactant, or we can use the other surfactant, for example, like SDS, sodium dodecine sulfate as surfactant, or sodium dodecine sulfate, the surfactant, or also we can use the treatment as hundreds. So it's it's depending on the lipid source and your application. So we can choose the different surfactant. Is it clear? Okay.
郭梦姐，他叫什么？
怎么了，吴刚？
他叫谁？
什么？
他的那个姓是怎么发音的？
So any other question, please?
Okay, Dr. Chen, I have some questions about some detailed applications. It's okay for you?
Yep. Yes.
Okay, I have first question is, do you have any data about the encapsulated efficiency? For example, mRNA or siRNA, because it's very popular in the worldwide. No.
So you mean the encapsulation, the efficiencies calculation?
Yes.
Yeah, we have done just some specific application for the encapsulation efficiencies. Yeah, I can check with my team to share that data. We have some data as I remember. So I will check with my team and can give you by email. So please give me emails. So.
Okay.
So yeah, so I can share it and check it with the team.
Okay. Third question is some factory were very concerned about how to remove the ethanol from the mixed solvent.
How to remove the ethanol? So the ethanol can be removed by the solvent, maybe just normal solvent extraction, or yeah, like a very common method like rotary vacuum extraction. You can remove the ethanol. It's very easy.
Okay, so the question is,
yeah.
Do we can provide the GMP equipment for production?
GMP production, actually, for GMP certificate, we are planning to provide it in 2022. Yeah. Not at this time, but we are planning to provide it in next year, 2022.
Okay.
Yeah.
So last question is about the scale of the parallel. Because FDA have two suggestions about the scalp. First suggestion is we call the structural scalp, and the second method is parallel. About the parallel scalp, there are many customers was very concerned about how to make sure every channel can maintain the same flow rate. Because
Okay. Actually, we have software, advanced software to measure the droplet size in life during with each parallel channels. So we can measure the droplet size for each channel. So it's the same size, means it can be it should be the same flow rate ratio between each channels. So we design the chip to make sure and test to make sure that before give to the customers. So every chip should have the same flow rate with every chip junction in parallels. So to check it, just using the microscope and determine the product droplet size, and we know exactly it's the same flow rate ratio or not. Because if different flow rate ratio, it can make different droplet size. So its final product, we will have the droplet size particle size or droplet size distribution. So you can know it's the same flow rate or not.
Okay.
Yeah.
Okay. Thank you.
Yeah, you're welcome. Any question, please? Okay, if you have more questions, not this time, so please feel free to email us. Feel free to email me or email us through the email address that we will give you in the chat box. So I will, yeah, I hope I will try to answer all of your questions through the email later. Yeah. So please, okay, can you see, everyone can see, yeah, the emails we already write down in the chat box?
Yes, okay.
Okay, yeah. So everyone, if you have more question and other question, please send us email. So if, okay, yeah. Okay, so thank you everyone for attending to our virtual workshop about microfluidic, and we hope we can see you again in the near future to support you. We have chance to support on reviews in near future. And yeah, thank you very much. Thank you, Bruce.
Okay.
Thank you everyone
Thank
from
you.
UA2 to support us with this my workshop. Thank you everyone.